want to celebrate Joanne's 7-year birthday this week? 1 week? 1 week? October 11th? I got a 0. 80. Remember what that was? The program, like the kids in the my grandparents watched it. I'm looking at it, God, they old.
So 80 years, she's saying. For those of you at home didn't hear what she said.
Oh, if you didn't hear me, I can always repeat it. I'm good at that. October 11th, I turned 80.
80.
Remember when 80 was old? Yeah, 39 was old. What, Jack Benny was 39 for like 40 years?
Yeah.
Yeah.
Then he forgot how old he was.
Thank you.
All right. We always thank the church for having this area for us to use and to be able to come in here. It supports the values of the church, and they support us, and we are very happy to be part of this DuPage Unitarian Church that gives us this emotional space, not only the physical space that we have to be with. So I think we're in our 14th year of Science Sundays. So yeah, it's very satisfying, challenging stuff. We were in isolation during the COVID times in the other room, and a lot of you watched online then, and some are still watching online now. And we encourage you to come in and participate because I think the conversations we have and the discussions we have are as important as the topics that Scott and I present. So, with all that, one topic near and dear to my heart is going to be today. But I do want to remind you, 2 weeks from today, we're going to have a very famous conservationist who happens to be my sister, Lois Winter, going to come in and talk about 50 years of being a conservationist. She started it out in high school. She applied for a program that was the ad in the paper in those days in the mid 70s said something like, "Boys, are you interested in working outdoors?" And so she showed my mom, who was a strict, staunch feminist at the time, showed her the ad, and my sister said, "Yeah, but I'm not a boy." And she said, "Haven't I taught you any better than that?" So she applied, and the Student Conservation Association accepted her. They made a program out in Mount Rainier, Washington, and one other place, and she'll know more about that. But anyways, I think it's she's been an inspiration for me. The things that she's done in terms of conservation and working with the National Park Service and many different national parks and so on. So I think it'd be a very interesting conversation. Today, Scott picked a topic near and dear to my heart, and I love hearing about it. So very happy to hear it from Scott's perspective as well too. So today, talking about the fallacies or the discussion points of the intelligent design people and where some of those maybe hit a few rocks in the way. So thanks a lot. Start out.
So those of you who are here, welcome to September 15th redo. You may recall last month, it was kind of a coin toss based on the weather. If it was going to be clear, then we would look at the sun and sunspots and talk about solar science and physics. And if it weren't clear, we were going to talk about impossible evolution. So I already had these slides made up. So I want to go ahead and talk about the impossible evolution idea today. And you'll understand what I mean by that as we go through the slides. Make sure my clicker is, there we go. Now I put a question mark after impossible because one of the persistent criticisms levied against the theory of evolution that has been resurfacing for 150 or so years is that it just can't possibly explain all the varieties of life that we see in the world around us. And, you know, these are good inquiries. If you see something that is perplexing or magnificent or aberrant with a living organism or a whole class or a genus, etc., then it's perfectly valid to say, how could this possibly have evolved, right? So those are good questions. Those are good scientific questions. And in the context of scientific questions, we form hypotheses, right? Those of you who have been around for at least a good portion of our 14 years or who have otherwise been fans of science know that there's this notion of a hypothesis. You put forward a hypothesis as an explanatory framework to say, why does X happen after Y? Or why is it that X has the specific behaviors or form that it has, right? You form some kind of hypothesis about why the world is the way it is, why something in the world acts the way it does. That's a hypothesis. And some hypotheses are borne out by experimentation and evidence, and other hypotheses find it challenging to be corroborated by evidence. The evidence does not support other hypotheses. So in this case, we have today two hypotheses that we're considering as an explanatory framework for the variety of life and different species in the world. And I want to specifically call out a few aspects of organisms that are perceived as being particularly challenging for that explanatory framework provided by the idea of natural selection. So Mike, and to a lesser extent I, through the years, have talked about evolution and genes and DNA and traits and haplogroups and on and on and on. We've talked a lot about the theory of evolution and some of its consequences, as well as talking about the genetic basis of evolution. But I really want to take seriously this second hypothesis, and it's called intelligent design. And intelligent design has as one of its keystones, cornerstones, the notion of what they deem, proponents deem as irreducible complexity. So this goes back to Reverend Paley, who in the, was it the 1700s, 1800s, early 1800s, said, you're walking along a beach and you find a watch or a clock, and all the complexity in that device is fully instantiated. Let's assume that it's perfectly working. Maybe somebody just dropped it there on the beach. And it's got all these gears and levers and springs and toggles and whatnot. And it all functions together elegantly and precisely to bring about the functionality of a watch. And what would happen if you took out just one of those cogs and springs and levers? Then most likely the watch would not function, or at least some portion, maybe the day tracker or the phase of the moon tracker if you've got a particularly complicated watch. But in the main, if you remove some piece of the watch, it won't work. And so Paley quite brilliantly said, organisms in the world must be similar. They have a similar level of complexity. If you take out my heart, I will stop working. If you take out my brain, I will stop working. You can take off a finger, I won't stop working, but I might get angry, right? But there are certain aspects of living things which seem to be a component, but an absolutely required component of the living thing, whether it's a plant or an animal. And how can you imagine these kinds of things existing if they didn't have a designer, an organizer, a crafter, a maker, right? And so William Paley put forth this idea in support of as an argument for God, for the existence of an intelligent designer. Now, let's take the religious aspect out of it and presume that an intelligent designer can be any intelligent, deliberate entity that is able to manipulate its environment. So we are intelligent designers. We make microchips and microphones and trillions of, well, maybe not trillions, but many, countless other devices. So we are intelligent designers. We design watches, right? And so you can suppose that there are other intelligent designers in the universe, maybe some aliens that help design the pyramids, right? Name your hypothesis. You can suppose that there is an intelligent agent behind it. Now, clearly, something of intelligence and facility built the pyramids. They didn't self assemble. But what about life, right? Why is life categorically different? Is it categorically different? Does it need designing? Does it need the action of a deliberate agent? So I talked about Paley's watch. Can Darwinism, so the theory of natural selection via evolution, can that explain everything about every living organism? If it can't, then if you look at the one of my favorite ways to look at evidence and accumulating evidence that supports a series of conclusions about the way the world works. We may call that belief, we may call it persuasion. One of the ways to accumulate enough confidence in a proposition that you support it. One of the ways you can believe something is you get evidence about it. You can get confirmatory evidence or you can get disconfirmatory evidence. And disconfirmatory evidence can be quite persuasive and powerful, right? Thinking about Newton's theory of gravity, the orbit of Mercury, according to Newton's laws, the orbit of Mercury should behave in a certain way. It's really close to the sun. That means it's in a relatively very strong gravitational field compared to the gravitational field of the sun out here at Earth and out by Neptune, etc. So if Newton's law of universal gravitation is universally applicable, it should apply to Mercury just as well as it does to Saturn and Earth. Well, when observations were made of the orbit of Mercury, they found that it did not, in fact, conform to the exact predictions of Newton's universal law of gravitation. So that was a scientific theory which had a point of failure, at least one, actually several, but that's the one I'll talk about, right? So you can find disconfirmatory evidence for a structure of hypotheses that have been largely validated by other evidence. We would call that a theory. You can find disconfirmatory evidence where a theory does not correctly apply to a particular context. And that's an invitation to make a new hypothesis. What does explain it? And why is this other theory or hypothesis incorrect? So that's part of what I want to talk about today, is that overarching notion of theory versus hypothesis and scientific validation. So the first thing we're going to talk about that has been put forth as a potential candidate as irreducible complexity is the eye. So this is protozoan, and it's a magnified illustration, so it's not an actual photograph. However, the picture on the right is a photograph, and the arrow there is pointing at a little sort of dark splotch there on this simple one-celled organism. And it turns out through various experiments, seeing how these organisms behave in different contexts where there is or is not light present, they have determined that this organism is light sensitive. And then they can do other experiments. They can look at the chemical composition of the dark patch, etc. But all it is a little patch within a single cell, right? This is not a complex organism. This is a single cell with different component pieces inside it, and it just has the most basic ability to differentiate light from not light. And imagine how that could be useful to the organism, especially if, I don't know, it's photosynthetic, right? Not meaning that it's artificially made by light, but rather that it feeds on light. It's a phototrope. It eats photons and water and carbon dioxide and makes sugars and other structures from it. All right, the whole process of photosynthesis. If an organism or a cell responds to light to get its nourishment, then it makes sense that that organism should be equipped with some means of detecting whether light is present or not. And even on simple single cells, there's this, is there light? Yes or no, right? And that simple sensor allows it to move variously toward or away from light. Maybe if you move towards the light, maybe you're going to be spotted by a predator. So maybe what your particular niche is such that you want to stay away from the light. So if you start swimming in ocean water and you get too close to the surface where it gets too bright, maybe it has a just like a little machine. Literally, it's a little machine that flips an internal status switch, and it causes the organism to move in a different direction. Right? So that's just a photosensitive spot. And then we can get into other more interesting developments. Well, what if you start making a little bit of a divot where that spot exists? If there's a divot, then you can begin differentiating the direction that light comes from, not just whether it is there or whether it isn't there. But if you're recessed into a divot, you can build sensitivity to light's directionality, and that improves your motility vis-a-vis light, right? You now know which direction to go other than just there's light, there's less light, right? So it enhances your motility and perhaps ultimately your survivability. And that's what Darwinian's hypothesis is all about. Or Darwin's hypothesis. And then, well, what happens if you start closing off the ends of that pit? Well, then you do something like a pinhole camera. You can start focusing an image. You get a clearer idea across that photosensitive spot. If you've got many different molecules that are acting as photoreceptors, and you get some that are bright, and some that are dim, and some that are even super bright, and some that are super dark, all within the same light-sensitive organelle, then you can start making out more details. You can differentiate darkness due to just global illumination, the sun is setting or rising, versus the shadow of a predator drifting above you or below you. So you can see that there's even more advantage to an organism that is able to focus light. But the problem with a pinhole camera is that one of the side effects is that it makes the light that you receive, it really reduces the amount of light. So it makes the light much dimmer, right? If you've ever used a pinhole camera, you get this very faint image and you have to be in really dark room or a dark box for it to work right. So how can we overcome that? Well, if we put a protective layer, a transparent layer of cells that's protective over it, well, that helps. But what if we, I don't know, what if somehow, like, there's just a bulge in the center of that protective layer of dead transparent cells? Oh my goodness, we've made a lens, right? So just a mutation that takes a protective layer of cells and means that sometimes it gets built with a bulge in it. Well, I'll be darned, those organisms survive a little better, right? Because now their pinhole can open wider and they can receive brighter light than if it were just a pinhole, and so on and so on. And many of these evolutionary steps occurred outside of a single-celled organism because they require many cells to collaborate, many photosensitive molecules in many cells to collaborate. And so you go from a photosensitive patch on a single organism to rods and cones in our eyes and in the eyes of other creatures. And I didn't think about this in advance, but one of the things that brings to mind is the mantis shrimp. I don't know if some of you recall that we've talked about mantis shrimp in the past. But you know how we have red, green, and blue, and there's some tetrachromes in humans that have 4 different light frequency distinguishing detectors for color vision. So most people are trichromes. There are a few human beings that are tetrachromes that have four. A mantis shrimp, and I forget the number, and this is just extemporaneous, so don't hold me to it, has something like 20 different individual colors that it detects. It has, instead of 3 separate photosensitive cones, color vision cones, it may have something like 20. Why? Well, that's a good question. And one of the hypotheses that's been put forward to explain that is that a lot of the processing that we and other complex organisms do to make sense of the world visually happens in our brain, right? We do edge detection, we do color separation, we do pattern detection. There are neurons that look for vertical patterns and horizontal patterns and color splotches and dim versus bright. All of this neural processing happens in our brain once the signal from our eyes reaches our brain. Well, what happens if you have a teensy little shrimp brain, right? Your teensy little shrimp brain doesn't have a lot of neurons to spare to do visual processing. So the processing has to happen in the eye. So the eye itself has to be sensitive enough to get information about the environment that might otherwise be delegated to image processing hardware in the brain. So interesting, the evolution of eyes. The key point is that although this was one of the earliest watchmaker kinds of intelligent design hypotheses levied against the theory of evolution by natural selection, it did not stand up to scrutiny. The theory of evolution can indeed successfully explain how eyesight arose, and indeed how eyeballs, the complexity of an eyeball with the sclera and the iris and the lens and the retina and the veins and the optic nerves that are on the wrong side of the retina. You know about that, right? In most organisms, most invertebrates, the optic nerve, which doesn't do anything to actually receive light, the optic nerve is scaffolded on top of the retina so that it interferes with light. Fortunately, there's a higher organism on the planet that has evolved a much more elegantly designed eye, and that would be squid and octopi and other mollusks. So they have better eyes than we vertebrates. They're what? 4 kinds of receptor. In what, the mantis shrimp? Oh, 12. Yeah, 12. Thank you, not 20. Thank you for looking that up. I was giving them too much credit. They're only massively more interesting than us. Mantis shrimp have better eyes than we do. I don't know. So do octopi in some sense. So the whole idea that we're the pinnacle of evolution, that's another argument for another day. Okay, let's talk about boundaries. This is one of my favorite topics in all of science, and I don't talk about it very often because it's at such a foundational level that I think a lot of times we miss out on just how relevant this notion is. There's you, and there's everything else. There's things that happen inside of you, and then there's things that happen between you and 13.8 billion light years away, right? And in order to differentiate things that happen in you from things that happen in the wider universe, you need some kind of boundary. Every living thing is encased within a boundary. So, I don't know, these Star Trek notions of intelligent clouds or energy balls that float through space, right? It's not going to work, right? You need a boundary that separates you from the environment. And a boundary has many great benefits. For instance, it allows for information processing across the boundary. You can differentiate what's happening inside from what's happening outside. Also metabolism. You can take material inside and you can process it and eject waste to the outside. There's all kinds of things that accrue from having a boundary. And this is such an innovative idea that somewhere around the 1600s, political communities of people decided that boundaries were a good idea. And we had the Treaty of Westphalia And that's when the very modern notion of nation states with fixed boundaries came into existence. So the whole notion of a boundary is a useful concept, and evolution has made use of this. It's not an amorphous thing like the ocean. I mean, the ocean is bounded by the seafloor and by the shore and whatnot, but life has a kind of boundary that allows for a differentiation of things that happen inside versus outside. Well, one of the things that happens inside a cell is that sometimes cells have to move. Now, an amoeba, for example, has the most fascinating way of moving. Some of you probably already know this, but an amoeba takes part of its cytoplasm, so that's all the gunk inside the boundary, right? Not counting the organelles. And it takes its cytoplasm and it builds these little microtubules of strands or beams or rods of little molecules connected together to make sticks. And think of it being like you're inside of a bag, and how do you move? Well, you move your sticks, and you move the bag with you, right? Well, that's how a lot of single-celled organisms move, is they form these microtubules within themselves, within the body of the cell itself, and it allows it to creep and crawl, right? Wonderful innovation. Okay, so why don't we take some of these sticks and stick them outside the boundary? And now what can I do? Well, then I can attach proteins to them on the part that's inside of me, and maybe if I put some extra protons into these proteins bound to the other side, then these proteins kind of curl up, right? Some proteins curl up, other proteins unwind when exposed to different chemical gradients, word for the day. And when a protein curls up or uncurls in response to a gradient, well, maybe the little microtubule you've put outside of your body moves. Well, suddenly you've got feet, right? A single-celled organism with feet, right? And so it's an innovation about motility inside the cell that can be extended beyond the boundary to even improve even more its mobility, right? So you can see how this innovation that helped a cell purely from the inside, like walking inside a bag or walking inside a balloon, if you extend it to the outside, suddenly you've got legs that you can put on the ground, and you can use the friction between your leg and the ground to propel yourself forward. So a wonderful innovation. And then it gets really, really weird. This is amazing. So movement, I've just talked about this. Amoeba, tubules in their cytoplasm. Oh yeah, viruses. I'm glad somebody wrote this up here. Viruses have a syringe, all right? So viruses are these stupid, I mean, as stupid as life can get. I don't know, some people think they're not even life, right? They're just these little packages of DNA that float around. And they have binding proteins that bind to particular places on other cells of other organisms. Most viruses are benign to human beings. They don't give a darn about our cells. But anyway, you get these viruses that float around, and they find a cell that they can grab onto. And then, seriously, they have a syringe, and they poke that syringe through the boundary of the target cell, and then they go, and they squirt out some DNA into their target, and the target takes up that DNA and says, oh, I know, I happen to have a DNA factory. That's what I am. And, well, here's some new programming code. I can make that. Sure, of course I can make that. And so it starts making more virus particles. And then eventually it makes so many that the target cell explodes, and suddenly you have a bunch of viruses floating around loose, much more than the one that infected it originally. And that's how we get sick, one of the ways we get sick. So anyway, viruses have evolved this notion of a stick or a tube to create a syringe, all right? And they have a chemical pump. So one of the things that happens once you've poked the target, you've got to inject it. So they literally have Again, through the contraction of individual protein molecules, they have developed an ability to squeeze, like squeezing a bulb on a turkey baster, right? They squeeze the bulb and force out the material. It's all driven by proton gradients. Myosin's the walking protein. I don't know if I ended up doing much with that. And then eventually it leads to rotary engines. So you take these tubules that are inside a cell. By the way, these are also implicated in the process of mitosis. So you've got centrioles in a chromosome, and a centriole is kind of where these microtubules grab on during cell division, and they help separate out the two halves of the chromosome so that one eventually makes a daughter chromosome. You always have pairs of chromosomes in a healthy living organism. You'll have pairs of chromosomes. And so during mitosis, the chromosomes split apart from each other, and the centrioles are a place for these microtubules. Again, microtubules. Nature reuses the same invention over and over again, right? To pull apart the chromosomes. All right, and there's some slight difference between the molecular structure of bacterial microtubules versus that found in eukaryotic cells. The $10 word eukaryote just means a complex cell with organelles. So it has a nucleus. That's its main differentiating aspect, is it has a nucleus that typically stores the DNA and whatnot. So if you look at the microtubules manufactured by bacteria and archaea versus the microtubules manufactured by eukaryotic cells, then perhaps not surprisingly, the microtubules manufactured by eukaryotic cells are a little larger and a little more complex molecularly, right? Not surprising. Eukaryotic complex cells are way down the line in the context of evolution. They appeared something like, 2 billion years ago. For the first 2 billion years of life on Earth, there were nothing but bacteria and archaea. So it took a long time for the first complex cell to arise. And I think Mike's talked a little bit about that magnificent innovation of evolution in the past. All right, viruses. Here's some pictures of some viruses. They look kind of like lunar landers. By the way, if you have a physical design that works in the context of some natural system, that physical design is probably going to be appropriate in other contexts as well. So a syringe is a syringe is a syringe. A thing that lands on its legs is a thing that lands on its legs is a thing that lands on its legs, whether it's a microscopic virus or a lunar lander or a cockroach, right? So recapitulation of physically relevant and chemically relevant processes is another aspect of nature, right? Things reuse the same design template, not because there's the same designer behind all of them, but because what works, works, and what doesn't work fails. And if it fails to reproduce. So that's, again, consistent with the ideas of natural selection. All right, so you've got this what's called a type 3 secretory system in viruses. So the little pump I talked about, where the individual molecules are powered by proteins, and they cause this pressure gradient. They mechanically pump. This is the way that pump works when magnified. Right? These are you can think of the individual bumps and little pellets or little things you can differentiate with your eye as bumps and whatnot. Those are individual molecules that go into building this engine, literally this pump. And then if you start looking at the flagellum, well, you take one of these spiky things that's hanging outside that you might be able to just sort of do this to. Well, what if you can spin it, right? Well, you take that pump, the secretory pump, and you can evolve the physical structure of that by tacking on a few more molecules, and you literally get a motor that is driven by a proton gradient. You start feeding protons across creating a potential difference across a physical space. You create an electrical gradient. This sucker spins. This is a mechanical electrical engine in a cell. Amazing. And a given cell may have many of these, right? Evolution is so innovative. Or is it an innovative designer, right? Well, when we look, scientists haven't definitively figured out the family tree yet, because if you've got a bunch of flagella that maybe just jerk back and forth a little bit, maybe that's helpful, but you're not going to be nearly as good in the race for survival as something that actually whips around and provides you a propeller. So the intermediate stages where you go from just being able to sweep back and forth like a little needle out in the water into something that maybe oscillates a little bit to something that twirls, well, those little oscillators, they got eaten. They're not around anymore. They got outcompeted, right? So species go extinct. Intermediate forms are often supplanted in a given niche by forms that have a superior compatibility with the niche in which they find themselves. Or the niche may change, and so the niche you occupied is no longer available, and then you die out, right? All kinds of dynamic processes go into making natural selection help. Could it technically have been designed? Could somebody sit down and write out how all these molecules go together and form a piston or a rotary engine? Yeah, somebody smarter than any living scientist, because this was discovered. It wasn't invented by a human. This was discovered. Is it plausible that somebody who knows how proteins fold and how proton gradients work and how membranes work and has a perfect understanding of that, could they design something like this? Yes. But why introduce that concept, right? It was wasn't Lavoisier. It was the French mathematician. It wasn't Leibniz. His name is escaping me. He traveled around with Napoleon. Starts with an L. I'm sorry? Lamont? No. Lemaitre? No. Anyway, he traveled around with Napoleon, and Napoleon apparently once asked him, he said, hey, do you believe in God? And the scientist said, maybe it was Leibniz. Anyway, the scientist said, in French, I can't do French, I have no need for that hypothesis. Right? Well, the same thing. Yeah, could you imagine that this could be designed? If we went to an alien world and wanted to build a microbe with a flagellum from scratch and all we had was knowledge of how DNA and proteins worked, could we, in principle, design something like this? Yes, but why? We've got plenty of them here. We just, it's already been evolved through natural selection, right? Antoine Lavoisier. Thank you. So you were right. I'm sorry for poo-pooing your idea. All right. So if you can see, literally see with this slide, I hope, how you can take the molecular structure of a pump and how that is homologous in terms of visual and structural layout to the rotary engine, right? You can imagine that evolution could take one and evolve the other. Now, to be honest with you, I should point out that there is a subset of scientists who think that the evolutionary mechanism went the other way, that simple organisms started with the rotary motor and devolved, lost functionality. We've talked about that a little bit. Lost functionality and ended up with a syringe or a pump. But the majority apparently is persuaded that evolution went the other direction, that from pump to motor. All right. Bacterial flagellum, I'm not going to talk much more about that. I've only got a few minutes left. Here's an animation of the flagellum. By the way, if the motor turns one way, the flagellum provides motion, and if you turn the motor the other way, it essentially winds up and binds up and stops motion. So they can go from moving to stopping. And then when they stop, suddenly their momentum or the currents in the water around them orient them in a random direction, and then they start their motor again. And what you see, if you're watching the critter move, you see, right? You see it traversing its volume, looking for something, chemotaxis, whatever, looking for chemical gradients that indicate food. But just by using that one flagellum, they can go from moving to hitting the brakes with one engine, right? Amazing stuff. All right, the next thing that is supposedly irreducibly complex, clotting. So this, there'll be a test later. This is a diagram of human, I believe it's human, clotting factors in play to turn your blood, your platelets and your red blood cells into plasma and whatnot, to make it clot so that when you are pricked by a syringe, you don't bleed to death. Tiny little leak over, 20 years, you can't stop it. What do? You lose all your blood, or you make new blood faster than you lose blood. I don't know. There's lots of options. But it's really good that most vertebrates have all vertebrates have these clotting factors, and it's a very complex cascade of chemical and protein reactions. Right? This is an abbreviated diagram of how complex it is. And so, if you're a proponent of the hypothesis of intelligent design, you say, "Aha! Blood would not clot if it didn't have all of these happening in this specific chain reaction. So this had to be designed." Well, first, why make it so complex? I'm not a molecular biologist. I don't know organic chemistry really, but I don't know. It seems very complicated to get a pretty straightforward result. So maybe it's overly complex. That's one of the things you can expect from evolution, is that it takes what exists before and evolves a new capability. And maybe the pathway to that new capability involves a lot of intermediate stages that if you were designing it from scratch, you wouldn't use, right? Anyway, it's very complex. So is it irreducibly complex? Well, if you look, fish, by the way, are some of the earliest vertebrate, right? Once the very notion of a chordate, thing with a spinal cord, a nerve cord running down their back, once the very notion of a chordate appeared on planet Earth, the very form of a chordate appeared on Earth, fish were a relatively early type of vertebrate, right? Long before land animals. So if you want to look for a primitive biomechanical process that exists in the world, you might want to look in fish, right? They're primitive organisms, or fish have within the whole genus, I don't know how that, within the whole genus, it has primitive exemplars. Well, here's a pufferfish whose blood clots, but it lacks 3 of the factors shown on that prior diagram, right? Well, how does it get away with that? This is complex. It's designed for a purpose. These guys are cheating, right? It works, right? So they lack 3 of the clotting factors. And then you go way back in the family tree, and there's scientists that have dedicated their lives to unraveling their larval phase of life. As you can think of them as little tadpoles, once again, I'm forgetting, embryonic stage, right? Post-embryonic, they're viable out in the world. And they're these little swimmy things, and they swim around and they look for a place to anchor, and they stick onto this place to anchor, and then they spend the rest of their life there. And you know what happens to their nervous system once they do that? It's dissolved and reabsorbed. They don't need it anymore. They don't need to think about anything. They don't need to contemplate the universe. They don't need to go anywhere. They found a perfectly good home, and they stick there for the rest of their life, and they get rid of their nervous system and their notochord, their spinal cord. Strangely enough, they've got some proteins manifest in their genome and turned in. They've got some genes manifest in their DNA that are instantiated as proteins, and these proteins are what we would call clotting factors in higher order animals. So these same proteins and this same genetic sequence that eventually gave rise to clotting factors is present in these, but they have nothing to do with clotting. These are more plant-like than they are animal-like. So nature has this idea of hold what you have that works and modify it. And it also has this wonderful capability of duplicating genes. Right? You got a gene that's expressed, I don't know, umpty squat times. That's a large number. And eventually some of those genes mutate, right? And so it gives rise to new structures and new behaviors and new capabilities. So replicate and modify. All right, so this is the last one I want to talk about today. This kind of gets off of the idea of irreducible complexity, but it's an argument put forward by those who support the notion of intelligent design. If you look at a human being and you count the number of chromosomes we have 23 and me, right? 23. If you look at a simian, an ape, a chimpanzee, a bonobo, a baboon, they have 24. How can we possibly be related to one another if we have completely different numbers of chromosomes? We must have a completely different design, right? Our genetic code is so different. Well, not so much. Chromosome number 2, up there in the picture. You know, I was talking about those centrosomes in a chromosome. It's a part where those little microtubules during mitosis, they grab hold and spread out from, or in, I don't know which direction it goes. But the centrosomes are where the little microtubules grab and pull apart during cell division. Well, it turns out that chromosome number 2 in every human being has 2 centrosomes. It's supposed to be centrosome. It's in the center. There's one of them. Chromosome number 2 has 2. It's stupid design. One of them is deprecated. It doesn't even function anymore. So why is it there? You may also have heard of this notion of telomeres. Telomeres are these things at the end of a chromosome that are implicated in how often the chromosome has been replicated. So senescence and cell death and whatnot. I don't want to get too grim about it, but every time the cell replicates, essentially a little cap of the telomere breaks off. And the next time it replicates, another cap breaks off. And eventually there's no more telomere cap on your chromosomes. And that's bad. For individuals in terms of our lifespans. But if you look at chromosome number 2, these telomeres, which are otherwise always at the end of a chromosome, are smack in the middle. Stupid design. Why would you do that? Right? Well, you know how this makes sense? If you had 2 chromosomes that stuck together and remain stuck together throughout the course of subsequent evolutionary processes. And we now have persuasive evidence that that's exactly what happened. And we are so human-centric in our view of the world. You know what we did after this happened, after this was discovered? When looking at the chromosomal structure of apes, they have 24 chromosomes. Do you think they're numbered 1, 2, 3, 4, 5 through 24? Nope. They're labeled 1, 2A, and 2B, and 3, 4, 5, right? Right? Instead of calling our chromosome 2/3, right, which many more species have that pattern, so we're actually chromosome 2 and 3 welded together. That'd be a good name for our chromosome. No. We are chromosome 2, and these guys are 2A and 2B. Right? Evidence in the natural world for evolutionary processes that does not bolster the notion of intelligent design. So we start with 2 hypotheses, and I've only touched on a few of some of the more widespread and well-known. And one of these hypotheses fails the test of evidence, so we end up with not 2 theories, but 1 theory, one scientific theory. Have multiple hypotheses, but only one of them stands the test of evidence and becomes a codified scientific theory. And the U.S. courts several times have agreed that there is only one theory that is an explanatory framework for the existence and variety of life on this planet. Most recently in Dover, Pennsylvania in 2005. And this book is written by one of the expert witness panelists who served in that trial. And many of the same concepts are covered in this book, The Greatest Show on Earth, probably one of the best books Richard Dawkins has ever written for explaining evolution, why, how, and how convincing the story is. And that's all I had for today. So I'll take some questions and comments. Thank you. Yep.
Is it? Isn't it possible that the creator didn't create individual things, but a system, so that the system then was basically evolution?
Mm-hmm. Yeah, so it's kind of a deist hypothesis that you get things started. Maybe you put the first replicating molecule. All it would take is a molecule that replicates and then becomes more and is subject to mutation. So if you get a molecule that replicates and is subject to mutation, you're off to the races. That's all you really need, right? Where did that molecule come from? How did life get its start? I wouldn't even call it life at that point. If a virus isn't alive, then this replicating molecule is not alive in some sense. We can argue about that. But yeah, you can. There's still an open question in science about how the first replicating molecule arose, right? So is it compatible with a deist notion that the first replicating molecule was designed and then we're off to the races? Yes, but again, using Occam's razor, if the natural world, another one of my favorite things is you get olivine, this natural magma solidifies into olivine, and olivine has been found on Mars and on Venus and on the Moon. It's just this natural granitic mineral that forms, and if you subject it to seawater, to salty water, it goes through a process called serpentinization, where it turns into serpentine. And in the process, it emits a current of photon, hydrogen ions, sort of bubbles off hydrogen gas as ions. And hydrogen, a flow of hydrogen ions is a current, right? And if you get a current flowing in a situation where you've got a molecular soup, that gives you the raw materials to make machines. And so you can imagine, and I may be way off base in terms of the way it really got started, but you can imagine a natural context in which replicating molecules thriving in a chemical and electrically potential rich environment started this process. So is it compatible with the notion of design to say that this molecule was placed here deliberately? Yes, but why? Why do you introduce that hypothesis? Especially if we know that it can happen through natural geochemical processes. And if there's a designer behind the scenes, then shouldn't we see evidence of the designer in more than just the starting gun? Right? All right, more questions?
Yeah, I wondered if you could tell us a little more
Could you hold it a
about,
little?
a little more about chromosome 21 duplicating and resulting in Down syndrome.
I can't. I don't know enough about that to speak about it extemporaneously.
You've made me want to look into it.
Okay, good. I know that what I do know sometimes is that the way new variations arrive in the genetic code is that a whole chromosome can be duplicated and passed on. If it's not particularly deleterious to the survival of an organism, then if it carries around an extra chromosome, it's got more raw material for potential future natural selection-based mutation. Yeah, right. I can hear it. Dave, is that mic on? Okay. Right. Right. And in 4 billion years, 4.2 billion years, we'll get humans. Yeah. Right. Yeah, there is this notion among advocates of intelligent design that the designer, let's call him God for lack of a better word, God puts guardrails, right? So the dinosaurs, yeah, that's not really the path I want. So let's get rid of the dinosaurs, right? So this notion that there's an intelligent designer that sort of every once in a while goes in and tweaks things, right? First of all, in the genetic and species record that we've uncovered on Earth, there's no sudden leaps in chemical or physical structure that can't be explained by gradual evolution. There are periods of accelerated evolution, but not whole cloth sudden changes, at least in organisms. Now, a meteorite's a sudden change. Did God throw that at the Earth? I don't know. I don't know why he had a grudge against the dinosaurs. But all right, another question. The other microphone seemed to work, didn't it? Oh, it died. Well, I'll repeat the question then. We have a question from online.
We have a question from online from Zombie Nature. The question is, how far back do the stuck together chromosomes go? I assume that he's talking about the chromosome 2 being stuck together.
So it's hypothesized that Neanderthals and Denisovans also had stuck together 2A and 2B. But the articles I read about that, I guess we don't have enough complete genetic material to yet determine whether Neanderthals and Denisovans, close relatives of humans. By the way, Neanderthals are no longer considered to be in our family tree. They're not ancestors. They're a co-evolved branch. But I don't think there's yet definitive evidence about where along the hominin family tree the chromosome merged together. Another question. We're getting the new batteries in the microphone here. Just give us a second. There are people watching online, and if you don't have a mic, they won't hear you. Like I said, if you don't have a mic, people online won't hear you. So, okay. So, what's your question? I'll repeat it. All right. So we're still evolving. What are we going to evolve to? Any projections? Yes, I have a projection. Humans will not go much further in terms of evolution. It's really hard for childbirth. We have all kinds of vestigial problems with our evolution, including in our behavior, our evolved behaviors with tribalism and violence and whatnot. My personal bet is that eventually, maybe not this decade, maybe not in the next 100 years, eventually we will create synthetic intelligences that are imbued with a notion of curiosity and exploration, and they will go out in the universe. So I kind of have a sci-fi view of it, but I don't think humans are going to evolve much more. Although Jim Walter and a few others who talk about evolutionary psychology will argue that in the past 10,000 years we've done artificial selection on ourselves for more pro-social behaviors and whatnot. And I think that's a pretty persuasive argument in some contexts. But where do I see us as a species evolving? I have no idea. And I don't think it's going to happen a lot. We do a good job of making sure people who have lower biological survival capabilities, we make sure that they can survive through things that aren't biological, like social welfare. All right, next.
I'm just thinking about the whole spine, rib cage, organs encased, legs, and how that design has been around for what would seem a long time. I know in the grand scheme of things, it's not all that long, but it seems to have held and become the winner of so many things, of so many species on Earth. Does that sometime go away and evolve into something different?
So there's this notion of the Cambrian explosion when all kinds of weird body shapes and plans appeared on Earth. We went in a relatively short period of 100 million years or so from free-swimming clumps of cells to critters and plants. And all kinds of interesting body plans, like creatures with spherical mouths and iron teeth and whatnot, which we don't see around anymore. Although I think there's a Komodo dragon might still have iron teeth, something like that. Anyway, read Your Inner Fish by Neil Shubin, and it talks about how a plausible scientific argument for how fish became tetrapods and eventually dinosaurs and mice and humans and whatnot. That Your Inner Fish.
As Mike and I have, both of us had comments on this in our programs. The original appears to be a species called Tiktaalik, which is an Aleut term, and it's physiologically fits every single part of us. 5 digits, the 2 bones in the lower arm, to 1 bone in the upper arm, to the shoulder, to the back, to the hip bones, to the leg bones. And it appears, and again, appearance only, that this is the prototype of all land animals, including ourselves and birds and lizards and frogs and everybody. So this, once again, is a marvelous example of the whole process, starting out with a single individual, which is extremely successful, the first animal to get on
Single
land,
species. Yeah.
and just exploding to cover the entire planet.
I wanted to add on that I thought that was a fascinating point you made about the body structure of animals and so on. But you have to recall there's a lot of animals out there. In fact, significant numbers of species that are not like that too. The whole arthropods, insects, clams, mollusks, octopi, so many other things as well too. So it is a very successful species, but there are many others too that maybe because of where we are and how we're constructed, we tend to think of that as the most successful. But I would say that bacteria, so many other species that are out there know how to handle us in a heartbeat, right? If it's a competition, right? They're there, and they showed us that with COVID. So it is a fascinating point, though, and I do think it's a very interesting topic to see how so many similarities, and it's a great support of the evolutionary frame that there are no real creatures in the vertebrate species that have more than 4 limbs. Right.
Which is an argument against dragons, by the way.
Exactly. That was my point. Yeah. Anytime you see a dragon, it's actually a freak of nature. It's impossible. It's a creation in the mind. And that's just the whole story of evolution. All humanity, all life really, is taking a running motor. It's like a motor that's been running for 4 billion years since life first came on Earth, and changing things piece by piece. It's like a Maserati driving down the highway and taking the doors off and putting a new motor in, doing all these different things to make a whole change and adjustment while the motor keeps running, right? That's life. The motor of life keeps running, and these changes are only changes that can occur while the motor keeps running. If you want to do something major and stop the motor, in most cases, that species or that individual at least doesn't survive. It's why the pharyngeal nerve in the giraffe goes all the way from the heart, all the way up around the hyoid bone and back down, and proceeds back something like 7 inches away from where it started, but it's 14 feet long. And there's an interesting YouTube of Richard Dawkins participating in a dissection of the pharyngeal nerve, and that used to work in fish. It just went right from
there.
the heart to the gills, right? But now in a giraffe, especially because of the long neck, it's a 14-foot long nerve that only travels a total of something like 7 inches. So.
All right, we are out of time. I'll stick around if anybody has any other questions or comments at the very end, but thank you, and we'll see you in a couple weeks.